Here it is. Here's the citric acid cycle. Now, appropriately, it is a cycle because everything, well, cycles through. You have neither a start nor an end. It just continues on and on and on. The initial substrate is acetyl-CoA, also known as acetyl coenzyme A, which is highlighted right here. Okay. Now this gives us a little bit of a flow chart for where we have been and where we're going. We started with glucose. We went through glycolysis and we generated pyruvate. We got 2 of them. Now, in the event that there is no oxygen present, you go through either homolactic fermentation or alcoholic fermentation. In the event that oxygen is present, well, you're going to have aerobic cellular respiration. This pyruvate is going to be able to enter the mitochondria, and with the help of acetyl coenzyme A, it's going to be able to enter the citric acid cycle. Now, this pyruvate will pass through this outer membrane right here through a channel called a pore ring. It's going to pass through this inner membrane here through a transport protein known as pyruvate translocase. Now, whenever we transport our pyruvate molecule into the mitochondrial matrix through that inner membrane, pyruvate is going to be converted to acetyl coenzyme A in a series of reactions. This is going to be catalyzed by pyruvate, or an enzyme known as pyruvate dehydrogenase. So effectively, we've got our pyruvate molecule. We've got 1, 2, and 3 carbons. Our transport protein, well, that's going to help the pyruvate enter the mitochondria. These steps 1, 2, and 3 are all a part of the pyruvate dehydrogenase enzyme complex. The very first thing is going to release one of our carbons. It's going to take this group right here and get rid of it. So we're going to have this decarboxylation take place very quickly. But have we formed our acetyl coenzyme A? At this point, we haven't. We have to use a couple of other steps in order to do that. Now, the pyruvate dehydrogenase enzyme is a large multi-enzyme structure. So if you remember when we talked about protein structure, we talked about the primary, secondary, tertiary, and quaternary structure. Well, the pyruvate dehydrogenase complex really illustrates the quaternary structure because it is a multi-enzyme complex, but it's working as one thing. Now, it contains at least 6 different enzymes in multiple copies. There are 2 regulatory enzymes, 1 with an unknown function, and 3 that are involved in the conversion of pyruvate. Now, these enzymes have awesome names: E1, E2, and E3. E1 is your pyruvate dehydrogenase, E2 is known as dihydrolipoyl transacetylase, and E3 is dihydrolipoyl dehydrogenase. The function of these enzymes. Well, just like we saw in the previous slide, the first thing that happens is pyruvate gets decarboxylated. So E1 actually carries out that decarboxylation. E2 catalyzes the transfer of an acetyl group to our CoAsh, which that is our coenzyme A before it's acetyl coenzyme A. Then E3 reoxidizes dihydrolipoamide, or lipoamide. Now, some of these things might appear like, I don't know, context? We're going to be getting into all these different components in just a moment. What I'd like you to know is, I'd like you to know the 3 different enzymes and the function of those 3 different enzymes. This E1, E2, and E3. So this right here, this whole slide, this whole display right here, is the pyruvate dehydrogenase complex as a whole. Now, what I'd like to do is I'd like to show you this right here is essentially E1, because our goal with E1 is to decarboxylate pyruvate. And, well, this is the pyruvate being decarboxylated. This right here is our E2, where the two carbons that remain from our pyruvate go through a couple of different steps and then are transferred onto the reduced form of coenzyme A, and we make acetyl coenzyme A right here, which it's just, it's displayed right there. Now, what you might notice about this is, well, where the heck is E3? E3 is everything right here. Okay, so what I'd like to do is walk you through some of the steps. So I'm going to erase all of my writing right here and just say what's happening. Here's our pyruvate molecule. Here's our enzyme. Here's a cofactor, TPP. Now, with the help of our enzyme and this cofactor, we're going to position pyruvate so that it can be decarboxylated. Okay, so what that means is that the three carbons of our pyruvate molecule right here are positioned so that they form a covalent bond with TPP, enabling a decarboxylation event to occur. Okay, so when our pyruvate has been decarboxylated, we form this compound or this molecule known as HETPP. That is our two carbons with a double bond and resonance stabilized. So we've got this resonance stabilized HETPP intermediate. So our coenzyme, or our cofactor here, is now bound to our two carbons that we really just need to continue going through this path. So then we see this molecule known as lipoic acid. That lipoic acid is a fatty acid which is permanently, or which is bound to our enzyme. Okay. Now what you'll notice here is we've got a sulfur-sulfur bond. So that sulfur-sulfur bond there is oxidized. Now what we need to do is we need to ultimately get these two carbons onto lipoic acid, which is done with an enzyme known as dihydrolipoyl transacetylase. So we get it through there. Then our enzyme is going to allow for some movement, some electrons, and deprotonation here, so that ultimately we have a compound known as acetyl lipoic acid. That acetyl lipoic acid makes these two carbons very easily removed from acetyl lipoic acid and transferred to CoAsh, so that we can take those two carbons and put them right here to make coenzyme A. Now the result of that, however, is the formation of dihydrolipoic acid. Well, dihydrolipoic acid is kind of ineffective. It's kind of not ineffective, it's pretty much useless. What we need to do is take that dihydrolipoic acid and return it to lipoic acid. So how are we going to do that? Well, we're going to do that with the help of some oxidation reduction reactions. This enzyme, dihydrolipoyl dehydrogenase, also known as your E3, is basically going to convert dihydrolipoic acid to lipoic acid. With the help of NAD+, FADH2, we're going to be able to make or regenerate lipoic acid. All right, now that's a lot to consume. What I want you to take away from this is E1 decarboxylates. E2 generates acetyl CoA, and E3 is going to regenerate lipoic acid. Now, one of the ways that I've always thought about this is if your aim was only to make acetyl-CoA, you only need E1 and E2. But if you want this to continue on and be functional, you need that E3 because E3 is going to do the oxidation reduction reactions so that you can regenerate lipoic acid from, let me add this in there, dihydrolipoic acid. I would say these are some of the key points of, these are the most important takeaways for the pyruvate dehydrogenase complex. All right, I hope that was helpful.